So thank you for Cameron and for the Society for inviting me to chair this panel discussion for the first session of this event. So my name is Duc H, and I'm the chief editor for EBio Medicine, which is a collaborative journal between the Lancet and Cell Press. So our editorial teams are split in two. So myself and Dan, my colleague Dan Stuckey, who is also in the audience. We are based in London Wall with the Lancet office. And that's the other half of the editorial team is based in Boston at the Cell Press office in the US. So the idea of our journal, EBio Medicine, is an online translational research that bridging the basic research and the clinical research. And I feel that our mission is very resonating very well with the mission of this event. So to develop new drugs and moving them through the translational research into the end user, the clinical end. So throughout the morning, we've seen 7 great talks highlighting the burdens of the NTDs and also emerging threat of drug resistance and our depleted pipeline for drugs. Also the poorly funded area in this important area. And we've seen also the difficulties in moving drugs from drug development and discovery into through the clinical trials to the end users. We see the laborious screening process of millions of drug candidates, compounds, through the in vitro assays, through the in vivo animal models, and through every steps we lose a large number of candidates, and we end up with only a handful of candidates to test in animals and also moving to phase 1 human studies. And sometimes we hit a brick wall with unacceptable toxicities or low bioavailabilities, or there's difficulty in translating animal findings into human findings, or a lack of animal models. Also, when you reach to phase 2, 3, and 4, as highlighted in the plenary talks, it becomes very expensive, so the lack of funding. But we also see innovative solutions from chewable tablets for children to discovery of new analogs to improve the bioavailability of our compounds. But especially in collaborating, I think the word collaborating is repeated many times in every talk. So through collaborations, we can resolve many issues. From sharing data, sharing compounds, to collaborative funding, working together, philanthropy work like donating drugs from big pharmas like GSK and J&J to MDI programs. And so I would like to open the floor for discussion to the audience, and I hope for a fruitful and lively discussion with our panelists. So there'll be microphones at the end of this, at the two sides of the audience. So if you raise your hands, if you have questions, microphone will come to you, I believe. Okay, so I'll give a minute or two for if you think of questions.
Can I ask a question?
Sure.
Probably supposed to be to give answers, but I like to answer a question and ask a question too of Professor Sattelle and the whipworm, which is very interesting. And you mentioned at the end that you were sending also samples, I think, for testing against other, the new compounds against other parasites, which I assume include ascaris and hookworm. I mean, you don't need to mention anything that's confidential, but I was wondering, do you think it would be good that it would be effective against other SDH species, or do you think it would be preferred that we have a specific anti-trichuris drug, given that resistance may be coming and things like that? So.
No, it's an interesting question. I think the colleagues we've sent them out to probably don't cover all those soil transmitted nematode parasites, but do cover a number of species, schistosomes, Couperia, and and so on. And we're very grateful for the collaborators looking at species where they have particular expertise. But certainly it would be interesting to increase that net. I mean, in view of the unmet clinical need, I mean, ideally it would be nice to have something that would hit all the big three.
Yes.
But perhaps with the success that Jennifer Kaiser and colleagues have had by combining different drugs, perhaps a drug that if it does turn out to be exclusive to whipworm, it perhaps could be combined with something that will hit the other two key players. Now I'm sure there are people here who have much better ideas on the practical implications of that, but at the moment we haven't any evidence yet that other parasites could be treated by this. It's early days.
Thanks.
I'm Dr. H from the University of Surrey. I had a question for Kelly Johnston. I mean, it was a really, really amazing tour de force in terms of drug screening. But one of the potential targets you mentioned was short-term high-dose treatment with rifampicin.
Yeah.
I just wondered if you could comment on how that might be applied in areas where there are latent mycobacterial infections, such as Leprosy or Borreliosa, or even tuberculosis, where there's a danger of resistance, as we heard in one of the later talks.
Well, there has already been other studies done by other groups that have shown that you can develop resistance in tuberculosis in treatments less than 2 weeks. So we're talking about 7 days treatment. Obviously, yes, these things would have to be looked into before it was rolled out. This is perhaps more of a test and treat strategy that could be implemented. But we're talking about something that hits the 7-day TPP, which is really what we're always striving for. Because although we currently have a macrofluoricide that could be implemented, we lack that TPP, doxycycline.
There's a question over there.
Thank you very much. Christian Boris from the Public Health Institute. I have a question basically to the whole panel, but probably mainly to Baeten and Ivan Scandale, because they are also working downstream and not exclusively in the development, the research and development of drugs. Having worked also in screening, etc., I know that the pain you all are going through, and also have worked in molecular sciences, so I exactly know where you're coming from and how many times you have to repeat. But I have a heretical question, actually. Don't we have at the moment actually a relatively good situation in terms of pipeline for most of the diseases compared to like 15, 20 years ago, and we actually have a problem in implementing. At the very, very end, we have very good malaria drugs. We have the combination drugs, and if I go to DR Congo, or if you work in northern Nigeria, you still see single therapy. 80% of the patients don't get combination drugs. We are talking about the elimination of sleeping sickness. Besides of the political problems with hamper actually elimination, we also see that the drugs do not really get where the patients are, and doctors are not willing. It's not about being trained. They are not willing to diagnose the disease because they tell us we are getting tired of looking for very rare diseases. So don't we have an implementation problem rather that we also should discuss in this audience in terms of how do actually the drugs go there? How do we actually find out? And that's more a social science question. The mechanisms, the best mechanisms to get acceptance of new tools in the area where they are needed.
Yes, I think you're right. No, I think that's a very complex thing to tackle, but I think you're right, because if you look at only the 10 diseases that are on the list of the London Declaration, which includes not everything that needs to be treated clearly, of those 10, 9 of them actually get treatment donated. The only exception is guinea worm, Dracunculus, which is probably the one that will be soonest eliminated and probably eradicated for other reasons. So there are definitely drugs available because these 9 other diseases are treated with donations. Now, they're not all perfect, of course, and I think in Oncocerca we can do better, and people are looking at it. In STH we have good drugs, but I think we will need different ones and better ones soon. Again, because resistance, I think, is around the corner. It's useless, I think, denying that. So I agree. It's also, and maybe even mainly a question of getting the drugs to the right people. Otherwise, I'm not at all an expert in the whole access discussions. I have colleagues who are specialized in it, but from what I see happening, I would agree with you, yes. But that should not prevent us from looking for new drugs where it's needed, and not think it's only in the last mile that the problem is. I think we also need better drugs for a number of diseases.
I also wanted to say something. I would definitely agree that there is a huge gap in terms of implementation research, and very much in the field that there's a notion that the clinical data somehow gets implemented, and obviously that's a very complex issue, not only, with researchers, governments, ministries of health, but I think there is, a misconception that somehow things get implemented, and very much, I think, in terms of tertiary and secondary level hospital settings and LMICs in Africa, where I've worked extensively, I think that routine practices and procedures and the drugs that are available, nothing much has changed in a very long time. So I think it is very interesting to understand, why that is. And I think the causes are very complex. But I think it is going, we need to talk to, open dialogues to the global, the international funders, and why aren't certain diagnostic tests and drugs listed on, for example, the Global Fund or by PEPFAR. So I think that is one issue. Then in terms of how we actually implement drugs and get diagnostic tests to be used according to the latest findings of trial data, I think that's a very complex issue. And within the DREAM project, I very much find that having a background in clinical research, it is somehow the road less travelled, in the sense that I'm trying to use a methodology from implementation research, so social science, health economics, educational tools around important clinical questions with hard clinical endpoints. And I think, I don't know, I'd be interested to know if other people have seen or worked on other projects which are similar. But for me, at least, there seems to be a gap in that area. And as you're saying, I think in routine care, there is such a gulf between what we know are guidelines and what is used in practice.
Can I just comment as well, from an AWOL perspective? We have an affiliated programme at LSTM called Countdown, which is funded by DFID, and that's doing exactly that kind of thing, because we're constantly told you can't give doxycycline in communities, you can't do it. But actually what they're doing in Countdown is they're actually doing implementation research around implementing an Antibobackia compound and other compounds, and looking at the social science behind it, all of these things that we've just talked about, because you're right, there is a gap between the people that say we can do things and can't do things, and then the people that are us who are doing the screening going, we've got these compounds, why can't we use them? Mm-hmm. So yeah, I think it is a gap, but Countdown is one project that's trying to address that gap.
So I believe the gaps probably in the public health research, and probably need the more attention from the governments as well as WHO, I guess.
I think
if
so.
they mandate.
Yeah, and also from in-country, it has to come from the ministries of health.
That's right, yes. Individual countries will have
notes.
Different public health policies and
but also
need to influence that.
Yeah, times are hard because there's AMR and hepatitis C, so there's lots of competing agendas as well.
That's right, yes.
It's not straightforward.
Hello, it's Ben Makepeace from the University of Liverpool. I'll be talking later on vaccines. I've got a sort of semi-veterinary background, so I'm very interested in the potential new drugs for whipworm, and whether if they have broader activity against other nematodes. And we've been talking about, obviously for most of the diseases we're discussing here, there's by definition with NTDs really there's no market, and there's all the barriers then to actually getting a product through preclinical development and clinical trials. But if there's a possibility of a drug like ivermectin. I don't want to big it up too much, of course, at this point in time, because ivermectin was clearly, one of the biggest drug discoveries ever. But if it has activity in the veterinary field against veterinary nematodes, where there's a lot of problems with resistance to other drug classes, potentially the market's absolutely enormous.
Yeah.
And I mean, clearly you've thought about this, but I think there's a mixture of huge opportunities, but also perhaps quite some big risks as well around compounding exactly how it's going to be protected and who's leading the agenda around it in the future, because that veterinary market in Western countries is, as I say, absolutely enormous.
Yeah, and just comment on that. I mean, there's a terrific history of an awful lot of drugs used for controlling human parasites came from the veterinary world, and so I think that's a well-trodden path, and it's really important to point that out. And I think there are. I'm not very knowledgeable on this. I'm sure others will be, but I think there are. There are whipworms of veterinary interest in pigs and perhaps horses too, but it is only a little bit aware of that literature. But I have talked to colleagues from the animal health, pharma industry, and they say although those are not major targets for them, they are actually markets of interest, and so that it could be well worth sort of exploring both those opportunities together. Sort of One Health context. I think would be well worth exploring here.
Yeah. Absolutely, and I think although you're right, there are whipworms of veterinary interest. I think the several billion-dollar question literally is, if they have activity against some of the other nematodes, like say for instance homunculus or Lavania, these sort of GI nematodes, which are not in the large intestine. So I mean, there'll be issues around exactly where is the drug active in its sort of parent form, does it metabolize, where is it metabolized, where are the, what's the PK, PD in the gut. But if it does have wider activity, then it really could be a gold mine, but hopefully that could plough back into the NTD field and actually provide a sort of platform to get forward.
No, that's a very good point.
I think also we also looked at that while we were looking at one or two other newer compounds. And I think, and I don't know too much about the veterinary market, so correct me if I'm wrong. I think the conditions would be definitely, first of all, that the cost of goods would need to be low. I think it's a rather cheap market, certainly the cattle, sheep. I mean, I think in the acceptance, probably the companion animals, where, I mean, cats, dogs, where people sometimes want to spend more money than on their own health, is my impression. But I think there, and that's my question maybe to you, I'm told that often these animals are treated with a pill or a formulation that has more than one active, that is, I mean, different worms, maybe also ticks or lice or ectoparasites of some sort. So I think that, which doesn't mean it cannot be done, but that means that there needs to be also absence of interaction between the different compounds in that formulation, I think, if that's correct.
Yeah, I think for large animals, apart from horses, which are sort of cross into the, are they pets, are they working on they're both depending on the country, but for livestock, particularly for ruminants, your unit cost does need to be low because they're very large animals, and if you're working on a per kilogram basis, then anything that's over a certain cost is just not going to be viable. You're right that people are willing to spend a lot more on companion animals. And as you pointed out, we've been very sort of spoiled by the macrocyclic lactones, which have got that incredible broad spectrum activity against not only the nematodes but ectoparasites. And I mean, I guess we just don't know yet with this new class of compounds what the issue is there. I mean, perhaps surprisingly, the arthropods and the nematodes, they're in the same superphylum, so quite relatively closely related. So actually the nematodes are more closely related to arthropods than they are to the platyhelminths, the flukes and cestodes. So there are possibilities around drugs that, like ivermectin, that could act across nematodes and ectoparasites as well. But yeah, ivermectin in particular, I think, has just changed the playing field so much that ever since it was used for flowery diseases, it's set the bar so high that I think it's, for instance, for antibiotics, doxycycline, for instance, the response of some of the sort of major organisations in the field has been, we've already got ivermectin, we'll just give one dose a year, what's going to be better than that? And I think that's really been a barrier to taking forward alternatives, and there's a risk that, okay, there's not a lot of evidence yet that ivermectin resistance and florinelizose is a big issue in humans, but it would be very strange if it didn't emerge. There's already data suggesting it is emerging in some parts of it.
Do these ectoparasites have Wolbachia, by the way? These arthropods?
A lot of them do, but there are very few cases where the Wolbachia are acquired by an arthropod. So there are one or two exceptions, like bed bugs, for instance, have an opposite relationship, but they're not a veterinary problem, they're a human problem, of course.
The question, I guess, initially, perhaps for Benny, but anybody with experience in this area would be interested. So, Benny, the trial that you mentioned, that unfortunately you had to essentially conclude that it wasn't worth continuing development of flubenzol. In terms of the reasons for that, for example, from the oral dose, was that surprises in PK or surprises in terms of how the worm responds to expected plasma levels?
I think we were somewhat surprised by the PK after subcutaneous, although there had been evidence before with another formulation. We had expected it would respond better to oral formulation as well, and the plasma levels, by the way, of the oral formulation are much, much higher than what you get after subcutaneous. So your peak is probably, 20, 30, 50% lower than with oral, but it stays much longer. So it is, what I usually say is that it seems like with flubenzol, at least you don't kill the worm by hitting it hard 5 times with a hammer, but you need to push gently and long, if you will, and then you kill it. But then the long exposure is an issue from the tox perspective, because even though these exposures were low, nanogram levels after 6 months basically, and we probably don't need 6 months, but we were not sure whether we can shorten it because we only give 1 shot, so how low could we possibly go? But that would mean that it also stays months in certain organs, and so from a tox perspective it wasn't doable. Testicular tox was going to be a problem probably. So we had already embryo toxicity, which meant pregnant women were definitely out of the question. Women of childbearing age would be, need to be very carefully managed with a chronic exposure would be difficult to do. If you then would also ask men to say take measures for 3 or 4 months after the last dose, I think that's practically undoable. It would be possible in a trial, maybe, but in a marketplace or in an MDA situation, I think we felt it was not doable. And then one of the bigger things was the clastogenicity. So it is gene toxic, anogenic, and one of the metabolites is clastogenic. And that is often considered not to be threshold dependent, but even a very, very low dose could lead to anogenicity and clastogenicity, and therefore in the long run carcinogenicity. So it was everything combined that made it extremely difficult and impossible really to move on. Was that an answer to your question?
Yes, pretty much. I guess it was also partly related to, in terms of the modelling that you've done before the clinical trials, how much of a surprise was it that the oral dose didn't work? And was that really a PK effect?
Oh yes, you mean with the current flubenzol on the market? Is that what you mean?
I'm sorry, I maybe misunderstood. I thought you'd done a single dose oral in your...
No, we have not done any human trial, and we were our internal gene tox review committee as well as the FDA told us, given the clastogenicity, you're not going to go to volunteers at all. So your phase 1 would need to be in patients, which is not easy to do, phase 1 in patients, but it would give us efficacy data, so that would be the upside. But we've never done trials in humans. The only trial done in humans with flubenzol is a trial in Mexico in the 80s, which showed promise, but also toxicity. That was a parenteral formulation. And all the rest is the current flubendazole, which is hardly absorbed. For STH, I mean, which is like mebendazole, there's almost no plasma level, so that was a totally different matter.
I suppose more generally, I was wondering about other diseases and people's experience of going to man, and the understanding of whether you get if you're seeing failures or surprises, are they surprises of PK or are they surprises of how everyone's responding to plasma levels?
Yep.
Okay. That's another question.
More EJ from Pharmadex in London. Following that question, Dr. Ben, I wanted to ask if you were looking at the killer data that is going to make you stop taking a candidate forward. So if the carcinogenicity, given the fact is not concentration dependent, are you suggesting that this is one of the screens we should be brought very early on the drug discovery? Rather than being done late in the program?
I would certainly suggest with systemically available benzimidazoles, yes. Although it could still be a flubendazole specific thing, the clastogenicity that is because it is metabolite dependent and not, benzimidazoles have similar metabolites, but they're not exactly the same. So it's not because flubendazole has it that other benzimidazoles have it by definition, the clastogenicity. But I think for members of the family, it's not easy. It's not cheap. It's not expensive to do. It's not, and it's quick. So I think it can and should be done. Yes.
So I have questions for Dr. Ivan Scandale or Dr. Kelly Johnston about these interesting compounds, the Tylomac that kill Wolbachia. So what's the stage of this drug development? Is that stage phase 1, clinical trials? Or?
Do you want to go? So the compound is currently in development with AbbVie, and it will enter in phase 1, should enter in phase 1 later in the year.
Right, okay. And in healthy volunteers?
Yeah, in healthy volunteer, the phase 1 is planned to be conducted in the US in a phase 1 unit from AbbVie.
So the partners involved are the DNDi and AbbVie and also AWOL.
Yeah. So we are just we're partners to develop the compound. So all the early work was done by AWOL and AbbVie, and we enter in the collaboration for development.
Right. So you will be sort of principal investigator for the trials, for the clinical trials.
We are not defined exactly how that's going to happen, but yeah, we will definitely have use some of our clinical sites to test the molecule, but we have a collaboration with all the other partners, so they will be closely involved into that process.
Thank you very much for the panelists and the audience for the discussion.